Um, Welcome, everybody. We will now have an exciting session on biomarkers in aging. As you know, we've seen many very promising anti-aging treatments during the conference, but many speakers really emphasized the need for biomarkers of aging. And there are really, by now, many tools available. I will talk about epigenetic markers, others will talk about metabolites, transcriptomic data, and so on. So I will talk about biomarkers which are called epigenetic clocks. As you know, the word clock implies a very accurate measure of time, and that really singles out these epigenetic biomarkers. They are the most accurate molecular markers of aging. I'll talk about some of the consequences of epigenetic aging, but also about anti-aging treatments. So I felt it would be good to give a short history of epigenetic clocks to get a sense of where we came from and where we are heading. I think because epigenetic clocks are technology, I use the metaphor of the development of flight. Like for me, it's very amazing that you can have the first flight in 1903, and then airplanes were already used in the First World War. And of course, now we have the space shuttle. So this development took place over 80 years. But epigenetic clock technology really took place over a time span of 8 years. There has been rapid and explosive development in that time span. So in 2013, several people recognized that you can develop very accurate age estimators. For example, the multi-tissue age estimator that applies to all cell types and all sources of DNA. But in 2013, we did not know whether these epigenetic clocks actually measure biology. Are they just a marker such as the graying of hair, or do they capture biology that is interesting to aging researchers? By 2015, this question was answered, we really know that epigenetic clocks predict lifespan and health span. They relate to a host of age-related conditions. By 2018, we now know that one can develop novel epigenetic biomarkers that strongly predict health span, and they really capture morbidity in a wonderful way. And also, custom clocks have been developed for in vitro studies. Mouse clocks have been developed by others about measuring aging in animal models, and we've gained quite a lot of biological insights. What about the future? I hope that in 3 years or so we have something what I call the universal epigenetic clock, or a version of it, which really applies to most mammals. The new frontier is to measure epigenetic age in rats, in monkeys, in bats, in the naked mole rat, really across mammals. So just to revisit things, so the publication from 2013 described this multi-tissue DNA methylation age estimator that whose age estimate is a number in years, and it really correlates with the chronologic age of the tissue in all sources of DNA, from sorted neurons to complex tissues such as kidney and lung. In 2015, the first publications came out that demonstrated that the epigenetic age of blood predicts how long you lived. And so the way that was accomplished was by studying large cohorts where the investigators banked blood samples in the 1990s, and so there was 20 or 30 years of follow-up information available, and so we could just see, did epigenetic aging in the 1990s end up predicting how long the person lived? And so by now there's no more debate that pretty much all epigenetic clocks predict lifespan. But there are, of course, differences in terms of power. Some are better at it than others. Now it's 2018, and several people have published longitudinal studies. So, for example, for the Chianti cohorts, we had 3 blood measurements available. They were all spaced apart by 5 or 10 years. And so we could see whether a person whose epigenetic age, let's say in 1995, was young, whether he continued to have a younger epigenetic age than expected in the year 2000, and how did that, and that was preserved in 2015. So we have these longitudinal effects. These plots are called spaghetti plots, and you see each curve is a human being, and you see for most people there's an increasing trajectory. I mean, out of thousands of people, occasionally you see some craziness going on. For example, this person, his age was estimated to be over 100, and then he dropped all the way down. So there was either a measurement error, or it could have been some cancer event. Leukemia can lead to overestimates, So craziness happens. But don't look at the outliers. Overall, it's absolutely remarkable. I want to say for 98%, you see a continuity. Epigenetic age keeps going up. What we call age acceleration is this discrepancy between the methylation age and the chronologic age. And we and others have shown that this age acceleration is remarkably preserved across time. So for example, if somebody had an age acceleration that was positive in 95, then 15 years later, for example, so this is mean time between different waves of a study, and the age acceleration remained highly correlated. Another way of saying it is, if a baby has a age acceleration at birth, chances are 70 years later they still have positive age acceleration. And that just reflects that epigenetic aging is to a large extent under genetic control. Heritability maybe 40%. Another insight that has now been clearly established is that epigenetic clocks don't just measure aging, but actually they also measure development. So even in the original epigenetic, the multi-tissue clock, I showed that the clock applies from age 0 to 100, so there was this continuous readout. But these studies have now been refined. For example, if you take an iPS cell and measure its age, it starts out with minus 0.6 years. An iPS cell, in certain ways, is perfectly young. When you differentiate it into a neural precursor, the age already increases, and if you further differentiate the cell into a slightly more mature neuron, you start to get positive ages. Positive age indicates postnatal ages, And so similarly, Tom Rhee showed that human fetal retina samples exhibit very strong correlations between methylation age and chronologic age. When you carefully look at the x-axis, the chronologic age these are negative numbers, meaning these are prenatal samples, and we get correlations of 0.68. And so what does it show? The epigenetic clock relates to development. And finally, I show you some data from Dan Geschwin's lab, where we studied three-dimensional cortical spheroids, right? You can develop the three-dimensional brains in the dish. And again, the methylation age of these spheroids is highly correlated with the differentiation day of the culture, So yes, these clocks apply to in vitro studies of neurons. They relate to development. I want to briefly mention another clock, and there have been now a couple of publications where people used cord blood samples collected from babies to estimate the gestational week of the child, you know, and one observes very strong correlations as well, you know. So what does it tell us? I think the findings are profound for aging research, because really these epigenetic clocks link in a continuous fashion purposeful molecular processes related to development and tissue maintenance during life to then adverse consequences later in life, you know. It's this continuous readout connecting development to aging. I want to shift gears and talk about a novel biomarker which we don't call DNA methylation age, we rather call it pheno-age. Pheno-age stands for phenotypic age, which is just another word for biologic age. I just didn't want to use biologic age because everybody has a certain definition of it. So this was developed by Morgan Levine for blood methylation data, and it's based on 513 CpGs, and you measure those and you get another age estimate. But this new clock stands out in terms of its strong predictive relationship for morbidity. Just to give you a sense, I show you Kaplan-Meier curves, where the blue curve shows people whose phenotypic age acceleration is less than 10 years. So in certain ways their clock ticks slowly, and the green curve shows you individual whose clock ticks quickly, so they have an age acceleration of plus 10 years. And this is the percent of survival, and you get a sense there's a fairly good separation, you know. So yes, these clocks very much predict mortality, but look also at the conditions listed underneath, because this phenotypic age acceleration in blood also predicts your risk of coronary heart disease. They relate to your coexisting disease counts, these morbidity indices. They relate to your physical functioning, how fast you can walk, how fast you can run. They can predict your cancer risk, not in a clinically useful way, you know, but if you have age acceleration, you're at a slightly higher risk of developing cancer. What is remarkable about this finding is that, again, it's all blood, you know, and this predicts various cancers, lung cancer, breast cancer, again, not as a clinically useful biomarker. It also relates to dementia. If we measure the epigenetic age acceleration in blood, it relates to your cognitive functioning in the elderly, which is unbelievable, you know. As I mentioned, it's particularly exciting that these clocks also relate to various measures of health span, or conversely morbidity. Why? Because many clinical trials, of course, attempt to prolong health span. So I want to give you a reality check. These epigenetic clocks do not stand out in terms of lifespan prediction. Don't walk away thinking the great thing is that these clocks predict heart disease and so on. Why? Because we have on some level far better predictors of lifespan, right? So blood pressure is a very strong predictor of heart disease, or your smoking status, or various frailty measures, or lipid levels, or glucose levels. That's really not the purpose of these epigenetic clocks. Rather, the great advantage of these epigenetic clocks is that they must relate to at least one root cause of aging. And I use plural because I think many of us believe there are multiple root causes of aging, but there's no doubt that the epigenetic clocks relate at least to one. Why do we know it? Because they predict diseases. You know? And by their nature, because they're based on DNA methylation, they must be proximal to an innate aging process, one of the root causes. Above all, they can be applied to cells in a dish. If you have an in vitro study, you cannot measure the blood pressure of your cells, right? But you can measure the epigenetic age. And of course, methylation age can also be used as a phenotype, a so-called endophenotype in genetic studies. For example, you may know that these genetic studies, the GWAS study of mortality, have been very disappointing. We always keep finding the same gene, ApoE. But if you replace lifespan by methylation age, you suddenly find many genes and exciting genes, and I'll show you that later. I want to give you an illustration of the power of these biomarkers for in vitro studies. So here, Kenneth Raj has carried out an in vitro study in fibroblasts, and the orange bars depict the methylation age in immortalized cells, and the blue bars in just controls. And what do we see? Although these fibroblasts are immortalized, perfect telomere maintenance by TERT, you see that they continue to increase in methylation age. What do we learn? Immortalization does not stop the epigenetic clock, at least in these cell types. I want to discuss various anti-aging treatments that allogenic hematopoietic stem cell transplantation is a treatment used for treating leukemia. And interestingly, it completely resets the age of the recipient to that of the donor. So, for example, if you have a 50-year-old leukemia patient and you irradiate him and replace all of his cells, get then this stem cell transplant, and the transplant repopulates the blood cells, these cells will have an age that is that of the donor. And interestingly, that's a long-term effect. That carries on for the next 30 years, So interestingly, the stem cell niche of the recipient does not affect the methylation age of these cells, which is a striking finding. Another therapy, menopausal hormone therapy. As you know, there was a lot of attention to that for these therapies. Unfortunately, they have severe side effects, and so many women don't take it anymore. So we analyzed blood samples from women who took menopausal hormone therapy and also buccal epithelial cells. In blood, we found nothing. However, in the buccal epithelial cells, we found a very strong effect that women who took this menopausal hormone therapy, in certain ways, they have young cheeks, And the question is why? Turns out buccal cells contain a lot of estrogen receptors, so they can in certain ways sense the estrogen. By contrast, blood cells do not. And so they don't pick up this effect, Many of you think immediately of standard factors such as good diet, exercise, education, and other lifestyle factors. Do they keep us young? And the short answer is yes, they do. Everything you've ever learned that keeps you young, from eating fish, moderate alcohol consumption, all of that works. As we have shown in a large-scale study of 4,600 individuals. So here are the findings. To be very precise, there are multiple epigenetic clocks. We have a so-called extrinsic epigenetic clock, which is very related to the Hannum clock, and then the intrinsic epigenetic clock, which is very related to the multi-tissue clock. And what you see, everything I said, fish keeps you young, fruits and vegetables, moderate alcohol, education, income. It's disgusting that people who have a higher income age more slowly, Exercise keeps you young. There are some factors that speed up both clocks. Insulin, glucose, BMI, metabolic syndrome speeds up both clocks. And there's a weird finding that, which could easily be a false positive, but anyway, chicken seems to keep this one clock from ticking faster, so don't trust it until it's further validated, However, so all of this reinforces what we know. However, let's really drill deeper into the data. Let's really look at the correlation coefficients. Remember, these studies are based on thousands of individuals. But here I show you correlation coefficients. Take education. Fortunately, all of you are highly educated. So the good news is it's very protective, p-value 10 to the minus 10. However, when you look at the correlation, it's only minus 0.1. That's a very weak correlation. Remember, it's in blood, you know. So similarly, vegetable consumption measured by mean carotenoids, p-value 10 to the minus 9, but the correlation is only minus 0.13. So these things, yes, they will be beneficial, but they won't have a drastic effect, you know, on your aging. By contrast, what does have a drastic effect on aging are these so-called Yamanaka factors that can, these are transcription factors that can turn a cell into a stem cell. And without any exception, every data set I've ever looked at drastically reset the age of the primary cell to an embryonic state. I want to mention, I think I mentioned it, we have several epigenetic clocks. There are several epigenetic clocks in the literature, and they really have different properties. So, for example, the multi-tissue clock is not related to telomere length. By contrast, the new phenotypic age is related to telomere length. To predict time to death, again, the phenotypic age acceleration measure is far better than all the other clocks, you know. By contrast, when you study, for example, gestational age in the fetal retina, the multi-tissue predictor would be best. So be mindful of that there's not one epigenetic clock, it's really plural. In my lab, we have this future goal of developing this age estimator that will apply to all mammals. We just implemented step 1, which is the development of a methylation chip for all mammals. If you have your favorite animal model, may it be dogs, rats, and so on, it can be applied. And my goal is to have epigenetic clocks for 50 species by 2021. That's a project funded by the Paul G. Allen Frontiers Group. I'm not sure how much time is left. I'm about 4 minutes. Oh, it's important, okay, okay. There's somebody who will delay my talk, but okay. Let's revisit telomere length in great detail. So, first of all, when you look at leukocyte TRF, telomere length, versus age in the Framingham Heart study, you observe this negative correlation, minus 0.27. That's the famous telomere shortening effect. From the very same individuals, we also had methylation data, so we can just correlate their methylation age, and we get a much stronger correlation, 0.81. And this is not cherry-picked. In every data set you observe these great discrepancies. So these epigenetic clocks have a far stronger association with chronologic age. So here are many studies, also German cohorts, German researchers have shown, probably were the first to show that yes, epigenetic age acceleration does not relate to telomere length. No question about it. However, Ake Lu carried out a large genome-wide association studies of epigenetic aging rates in blood, right? The question is, are there certain SNPs that accelerate the epigenetic clock? And guess what she found? One of the major hits, p-value 10 to the minus 13, look at that, TERT locus, you know. The TERT locus, TERT is part of telomerase, which maintains telomere length. And here I show you GWAS findings from leukocyte telomere length. These are the blue dots versus GWAS findings from intrinsic epigenetic age acceleration, the red dots. Indistinguishable, you know. So although telomere length is not correlated with the epigenetic age, when you do a genetic studies, they both converge on TERT. However, there was a paradoxical finding. Namely, when you carefully looked at the genetic variants in this third locus that are associated with younger biological age according to the telomere clock, these genetic variants were associated with older biologic age according to the epigenetic clock, and vice versa. In other words, if you had won the genetic lottery and your parents gave you a genetic variant that predisposed you for longer telomeres, then you just had lost the genetic lottery with respect to the epigenetic clock. Paradoxical finding in some shape or form. But it's real, I can promise you that. We've validated it over and over and over again, even in unpublished studies. The finding is real, the interpretation requires debate. I want to end up mentions progeria. We have an unpublished but highly accurate clock for skin and fibroblast samples, and I applied them to fibroblast samples collected from Hutchinson's progeria individuals. And guess what? I find no age acceleration. I've worked very hard on that question, but I just don't see it. At least, so Hutchinson's Guilford progeria is not associated with epigenetic aging, at least in fibroblasts. Conversely, there are other progeria, the so-called Werner syndrome, which is an adult progeria, and that is associated with epigenetic age acceleration in blood, and also Down syndrome, which some clinicians think of as actually the ultimate progeria, that is also very strongly associated with age acceleration. As a matter of fact, if you ask me what conditions is associated with the strongest age acceleration, I would say it's Down syndrome. So in conclusion, we have now several epigenetic clocks. All of them correlate strongly with age. However, deviations from age, these deviations that we call age acceleration, they occur often in a tissue and cell type specific manner. Obesity accelerates aging in the liver, but maybe not in other tissues. Also, methylation age can apply to cell culture models and various ex vivo systems, that what makes it very exciting as a screening tool. Also, I want to emphasize, methylation age will not replace the standard clinical measures from blood pressure to lipid levels and so on. The epigenetic age at this point has no clinical significance because we don't have any treatment yet for reversing your epigenetic age. Having said this, I very much urge people who conduct clinical trials to measure methylation, especially when you do anti-aging clinical trials. However, you need to be judicious about the source of the DNA. Many people collect blood, but I think I've highlighted a case where it was very important to collect buccal epithelial cells, But also think of collecting urine. If you give me a cup of pee, I can actually tell you how old you are. In urine, there's enough DNA to do that, And so in any way, so I'll stop with that. Thank you very much.